Hello, everyone. I'm Nathan. I'm a PhD student in the Department of Statistics at UW-Madison in the Solis-Lemus lab. And I'm going to talk to you about a method we've been working on for inferring phylogenetic networks at scale. So as most of you probably know, one of the biggest issues with phylogenetic network inference is that it scales very poorly. Kevin talked about this at the end of the last session as well. And so these are some runtime figures for SNAC, a pseudo-likelihood method. And on the x-axis are different networks that get progressively more complex. And the y-axis is runtime in hours. And this is the previous version of SNAC, not the new one that Kevin was talking about. But simple 10 taxa networks take sometimes less than an hour, sometimes with 3 reticulations in really bad cases as much as a day. And by the time you get to 20 taxa with 3 reticulations, we get some runs that take over 100 hours and some runs that take close to 150 hours. So you can imagine by the time that you get to 30, 40, 50 taxa, it's just completely infeasible to use SNAC. And so that's the issue that we're trying to solve. And so the way that we're going about it is with a divide and conquer approach that starts with whatever input data you want, gene trees, multiple sequence alignments, biallelic data. You just need to be able to do 2 things with it. The first is you need to be able to infer a pairwise distance matrix. And the second is you need to be able to break down all of your input taxa into small disjoint subsets of taxa, and then infer small networks on those, preferably with something that's accurate, like a likelihood or pseudo-likelihood method. And then the last step of the little pipeline is to use the information that's in that distance matrix and in the smaller networks and merge them together into one large network on the whole set of input taxa. And there are methods that exist for every portion of this pipeline, a lot of methods, except for that last part, which is where our method that we call infinite comes in. And so infinite stands for infinitely scalable phylogenetic network inference. It's a little full of itself. But, and it sort of leverages the speed of neighbor joining, which is a very fast but inaccurate method, and the accuracy of more accurate likelihood-based methods. And so the way it does that is it does what neighbor joining does, where at each iteration, it merges the 2 most similar nodes in the sort of tree or the network that you build up from nothing. But it only considers nodes that are siblings in your constraint networks. So here we have a very simple case where we've inferred 1 4 taxa constraint network. Those can be much larger. They can preferably not be much smaller. And usually you're going to have a lot more than 1, but just for a simple example, there's only 1 here. And the only 2 siblings here are A and B, and then C and D. So those are the only 2 nodes that are considered for merging in the inferred network. And turns out that in the first iteration, A and B are more similar, so those 2 get merged together. And you can see the shortest path in the constraint network that connects A and B has the incoming portion of a reticulation. So in the inferred network, we place that half of the reticulation. And then the constraint network is simplified to reflect that A and B have been combined into this new node. And the other half of that reticulation is sort of left pointing to nothing. Then we repeat this in the next step. There should actually be 3 siblings here, but there's only 2 shown. And in this next step, C and D turn out to be the most similar. And again, the shortest path connecting those 2 in the constraint happens to have the other half of that reticulation. So now that reticulation is completely placed inside of the inferred network. And then only one option for the last step, which is to combine everything into the full network. And again, usually you're going to have many more constraint networks than this, so it's going to be a bit more complicated. And the actual inferred network is unrooted. It's just easier to draw this rooted. And so we have this method, and we want to know how well it does. So we did a simulation study that starts with simulating ground truth level 1 networks with the sci-fi network package in R, which is by Josh Justison, who had a recorded talk in the previous session. And we need those 2 things for the method, the distance matrix and the constraint networks. So for the simulation, the way that we got the distance matrix is we take the major tree of the ground truth network, and we calculate the pairwise internode distance matrix. And then for the constraint networks, we do subset decomposition with the method described in Sate 1. And for those constraint networks, we take those subsets of taxa and we prune their subnetwork off of the ground truth network. But that's a little boring. That's all ground truth data, basically. We want something a little harder. So we put error into the system with Gaussian noise into the distance matrix and random nearest neighbor interchange moves on the constraint networks, so we can see how well the method holds up to random noise. And then we can throw all of that into infinite and see how similar the inferred network is to the ground truth network that we simulated at the very beginning. And it turns out that it does pretty well. So these are some results for networks with 1,000 taxa and 100 replicates with reticulations from 1 to 30 reticulations. And the x-axis is the level of error in the constraint networks, binned rather than continuous, because we tried that, but the figures were not very legible. So low to very high, and then the colors of the bar are the distance matrix error, the level of error in the distance matrices from low to very high, and the y-axis is the error in the inferred network between the ground truth in hardwired cluster distance. And there's a very clear trend with each type of error, which is obviously the more error that you have in the pieces that you infer, the worse you're going to get, the worse the final product is going to be. But if you're able to infer both portions of this with very low error, you can get near zero error in your final inferred network, even if it's with up to 1,000 taxa. That is with one caveat, which is that the size of those smaller networks is very important. So this figure is, this time, only with low error in the constraint networks, but varying error in the distance matrices, and with a varying size of those smaller constraint networks. And so we see that as the constraint networks get smaller and smaller, you can imagine you're going to be missing some reticulations, and you're going to be relying more heavily on neighbor joining than you are on whatever your likelihood inference method is. And so your accuracy is going to get progressively worse, even when all of the underlying constraint networks are very well estimated. And we found that a nice sweet spot is around 25 taxa, where you see there's not much more increase as you go to 30 in accuracy. And that's where SNAC also starts to, and other pseudo-likelihood methods start to really drop off in their runtime and become infeasible after 25. And so the method itself is, the real bottleneck for speed is the inferring the networks. The method itself with up to 1,000 taxa takes less than a minute to run. So the real bottleneck is whatever pseudo-likelihood or likelihood method you use. And so then in summary, we can infer networks with up to 1,000 taxa accurately. We also have simulations with up to 2,500 taxa, but those take a long time, and so those are still running. We don't have figures for those. And the other caveat is that the constraint network size is important. The bigger, the better, but it's important. And as far as next steps go, you may have noticed if there are any reticulations between the small networks that you infer, there's no mechanism for sort of detecting reticulations or introgression or hybridizations or anything like that between the smaller networks. So we're working on figuring out a way to do that sort of efficiently within the algorithm as it is now. And also then statistical consistency, we already have some nice consistency results. It turns out the method is consistent if your network is level 1, but we just need to make those arguments a bit more robust. And so all of the simulation study and the method itself are written in Julia, except for the portions that use other packages, and are available as open source software on GitHub here. There's really no documentation yet. There's some very basic intro instructions, but it's in a usable state. So if you want to try it out and tell us how you feel about it, that would be very much appreciated. And finally, I'm just going to plug my advisor, who has a talk about networks that is not in the network symposium. It's in a different symposium for some reason, so just thought I'd mention that so you don't miss it. Thank you.
Even more than that?
Yeah, so it actually, in our simulations, it performs the same regardless of the number of reticulations, but that's probably an artifact of the fact that we only use level 1 networks, and if you have like 100 reticulations, for the network to be level 1, there's still, all of those reticulations are going to be very close to the bottom. And so they're going to be pretty easy for this to deal with. It really falls apart when there's reticulations higher up. Yeah. Or between distant species or taxa. Yeah.
You touched on something that's been bothering me, although it's not central to your question, and that is, why should noise be Gaussian?
There's no real reason why it should always be Gaussian. It was more or less just the convenient source of random noise. You could definitely.
No, maybe there's a good reason. I just don't know what it is.
No, here there's no good reason. Yeah, the main thing is Gaussian has a sigma, which is a value that we can really record and compare to. We can make solid statements about the amount of signal and noise in the distance matrix, but you can do that with any random noise. It's just convenient, is the short answer. Thank you. Thank you.
Hi everyone, I'm Thore from the Natural History Museum in Oslo, and I'm excited to present today one of my PhD projects. But first of all, please allow me to take one step back and tell you a brief story about Ernst Mayr and his interaction with Duvenbode's rifle bird. That bird was quite important for the career of Ernst Mayr because that bird was only known from 3 individuals only, and that was the reason why Ernst Mayr's career started, and he was actually sent to New Guinea to have an amazing time being the first ornithologist in the remotest parts of that island. But he was never able to find this bird, so it was a big failure of Ernst Mayr. So starting a talk with a failure of Ernst Mayr might be a bit weird, but it led to an interesting discovery because his PhD advisor examined that bird in more detail, and he found this is actually not a species. This is just a first-generation hybrid between the lesser Lophorina and the magnificent rifle bird, and led to the conclusion of Ernst Mayr that in general, hybridization between species can be rare and is quite unimportant. Well, today we know that is not true. Hybridization in animals is more common than previously thought. And here's another bird example of this interesting case where you not only have a hybrid between 2 species, but it's more complicated. This is the first of its kind wild triple species hybrid, and it's a mixture at different taxonomic levels. So it's both interspecific and intergeneric hybridization. You can find even more extreme examples. Well, this was, even though in captivity, a hybrid between a Russian sturgeon and an American paddlefish, resulting in both viable and fertile offsprings between species that diverged over 180 million years ago. So quite extreme. And while we have these examples that are more recent, genetic exchange among divergent groups has also taken place in the distant past. And we can see several examples, for example, between the Komodo dragon, Australian sand monitors about 11 to 5 million years ago, or between North American freshwater fish at least 20 million years ago, and a more extreme example between the ancestors of fungus gnats as early as 114 million years ago. So there is an increasing number of genomic studies that report ancient introgression. But this is a bit problematic because most of the methods developed for detecting hybridization were originally aimed for analysis at the level of populations or recently diverged species, for example, modern humans, Denisovians, Neanderthals, as shown in background here. But this is problematic when detecting gene flow between divergent groups. Why is that the case? In more divergent systems, the assumption of constant evolutionary rates is violated. Therefore, rate variation is both present and affecting these introgression tests. This is quite visible in a time tree of vertebrates when you just have a closer look at 2 random groups, for example, this chameleon and this giant tortoise. You can see that the length of these branches is not equally long. So why is that problematic when applying methods to detect gene flow? Well, let me first of all introduce you to some relevant statistic approaches and how they are used. Just to those of you that might be not aware of the useful and widely applied introgression test, the D-statistic, or also known as ABA-BABA test. So how to calculate the D-statistic? You have a 4-taxon phylogeny, P1, P2, P3, and P4. And the ABA-BABA test considers ancestral A and derived B alleles across the genomes of these 4 taxa. So, as visualized here. So without introgression, 2 particular allelic patterns, ABA and BABA, should occur equally frequent due to incomplete lineage sorting. In the case of introgression, you would expect an excess of either ABA or BABA, resulting in a D-statistic that is significantly different from 0 and indicative of gene flow between these 2 taxa. So this is basically resulting in this formula. It can be more simplified. D is the normalized difference in the sum of ABA and BABA across the genome. So sum of ABA minus the sum of BABA divided by the sum of ABA plus the sum of BABA. And then this formula is basically just summarizing that. But how is branch rate variation affecting the D-statistic? To study this in more detail, we simulated genomic data using MS Prime, and we used different settings for a 4-taxon phylogeny, again with P1, P2, P3, and P4, and the following parameters were defined. Different population sizes, different divergent times, 1 recombination rate, different mutation rates, also different rates of introgression between P2 and P3, and branch length variation was introduced by simulating different rate factors for the P2 branch. This was done for 10 to 50 replicates per setting, so resulting in a total genomic data set of more than 4,000 BCF files. When calculating the D-statistic and in the presence of introgression, so you can see on the left here 3 different divergent times with 4-taxon phylogeny for a given population size and a given mutation rate. So in case introgression was present and rate variation was absent, D-statistic was performing well. This was also the case when introgression was present and rate variation was present. So in that case, the D-statistic is a reliable indicator of introgression. However, in cases where you have no introgression at all, with the same settings and rate variation, we can find false positive signals indicated by these bold box plots on the right. So here we have false positive signals of gene flow due to rate variation. This is also the case if we consider parameters with a lower population size. We have again false positive signals, as you can see here, but also with a lower mutation rate, the same pattern, false positive signals in the presence of both rate variation and introgression. Also, if we don't have not only a lower rate, but also a higher rate of the P2 branch, we can see the same pattern, again, false positives. What about the reliability of other commonly applied methods that use gene trees? Therefore, I use the same simulated data set to infer gene trees by using IQ-tree. IQ-tree is a fast and effective stochastic algorithm to infer phylogenetic trees by maximum likelihood. And I focused on 4 different tests I will just briefly introduce here. The first one is D-tree. That's equivalent to the D-statistic, but based on frequencies of tree topologies. The second one is Kolbow, estimating introgression via branch lengths. Then SNAC, we already heard about during the session today, maximum likelihood inference of phylogenetic networks. And the last one is a method introduced by Maya Machina Salzburger in 2017, that's why we called it MMS17 method, that compares mean divergence times for all pairs of species within a species trio. So to summarize briefly the results of D-tree and SNAC when being applied to our simulated data set, you can see here that we have non-significant results, even though they were quite high for D3 when rate variation was present and introgression was absent. However, not a single significant result was obtained for D3 and SNAC, and therefore we can conclude that both D3 and SNAC are robust to rate variation. That looked quite different when we applied Quibble and the MMS17 method on our IQ3 trees. Here you can see that we have false positive and significant results in both cases. So both of these methods are sensitive to rate variation and produced false positives in the presence of varying rate. This led us to the idea to introduce a new method because in the presence of rate variation, patterns might occur caused by homoplasias that just mimic introgression and cause false positive signals, as you can see here indicated by randomly distributed ABBA patterns along the chromosome. However, when you have clusters of ABBA patterns along a chromosome, these should represent true patterns of introgression. So homoplasias should be randomly occurring, while true clusters should be introgression. We implemented this approach in the widely used software D-Suite, and I just want to show you how our new test was performing. Again, using the same simulated data set that I mentioned before, we developed two different versions of our test, both a sensitive version that is using all informative sites and a robust version that is using these strong ABBA sites that I just mentioned before only. And here you can see that our robust version is not affected by rate variation at all, and therefore robust to rate variation. In addition, I don't have the time to show you all the other simulations we did, but I just want to mention that the robust version is also not affected at all by among-site mutation rate variation, by recombination rate variation, or by incomplete lineage sorting. And this is opening the door for applying this robust ABBA site clustering to also some other data sets. So we have used that also for an empirical data set. In 2021, there was a comprehensive genomic data set for Tanganyika cichlids published, and there were 4 tribes identified that should be completely introgression-free. So there should be no genetic exchange between these cichlid tribes at all, as indicated by that study. However, when using our new ABBA site clustering, you can see the following plots here, where the detection of ABBA sites is indicated by changes in the slope. And I show you here 2 chromosomes of 23, and only in a single one we were able to identify a change in the slope when applying our ABBA site clustering test. And this change in the slope is indicating that there's an increased frequency of strong ABBA sites for this particular chromosome that might be caused by an inversion or a structural rearrangement in that case. So I just want to conclude that widely used methods for detecting gene flow actually are problematic when being applied for divergent groups. These statistics, Quibble and the MMS17 method, produce false positive signals for introgression due to branch rate variation. D3 and SNAC are reliable indicators of introgression in the presence of rate variation. And our new ABBA site clustering is both robust to rate variation and mutation rate variation and can be used when testing for introgression between divergent groups. So the take-home message is consider rate variation when applying statistical methods for detecting gene flow. And I would like to thank my advisor, Michael Matschina, our collaborator, Milan Malinsky, and Matt Hahn for useful discussions and advice. And if you want to have a closer look at our simulations, you can have a look at our paper published in Systematic Biology. Thanks. Questions, comments? Yes?
So I have 2 questions if we have time to go. The first one is they performed well in the presence of rate variation.
4 tree-based methods. So
4, SNAP.
D-tree, SNAC, Quibble, and the MMS17 method.
Okay, I meant topology versus branch lengths.
Okay.
But are you essentially showing that IQ tree correctly reconstructs the topology in the presence of rate variation?
Yes, I mean, we check the topology of the reconstructed gene trees when using IQ-tree, and that was the for the different parameters where we introduced different degrees of rate variation, so a rate for the P2 branch. IQ-tree was showing the correct topology, so we did a topology check.
Right. So, I mean, in some sense, what you're saying is IQ tree can build trees correctly under the conditions that you're describing. I mean, topology correctly. And so maybe a future thing to think about would be, are there conditions under which the trees tend to have errors?
Yeah, okay, yeah, maybe in future to check, yeah.
And then the other question I had was the clustering idea, which I think is very interesting. It seems like that would depend to some extent on recombination, and you might see challenging situations in species that have microchromosomes and macrochromosomes where there's, and is that something that you have any thoughts about?
Yes, that's true. Of course, we did not test something like that for extreme cases like micro chromosomes, but what we did, we used different alignment lengths for when building these gene trees. So 3 extremely different, yeah, and we got the same results, so it was not depending on alignment length. And yeah.
Thank you.
You're welcome.
Okay, let's see. So my collaborators, you've seen a few of those repeatedly already. Cecile and Hector are here, and Jinqing is not. But okay, so the broad question I'm trying to get people to think about is what features of an unknown, complicated species network have a chance of being inferred? So the technical word for that is identifiable, and I'm not going to define it carefully other than to say you should think of it meaning is theoretically you could infer that those features from data. If something is not identifiable, that's equally important. It means there's no way to extract it from the data. And I think that second question is maybe not been thought about enough, that there's going to be limits to what we can infer, and it's good to understand them more clearly. Okay, so the answer to that question, it's a very vague question, may depend on what we're willing to assume about our network. Is it level 1? Is it level K? These red properties are going to be ones that pop up later in the talk that I'll define when I get to them. Maybe the network needs to be outer labeled planar. It will depend on what you're trying to say. We saw a talk at the beginning of the first session here about how one could infer the tree of blobs of a network, even with no restriction on the structure within the blobs. If we assume other structure, maybe we can get the order in which things are joined around the blobs. Maybe we could get the full structure of the network, but that just may simply not be possible. It also is going to depend on the model of gene tree formation. So we've heard about ILS. And when I say ILS, I mean the completely independent multi-species coalescent. So all lineages behave independently of one another. We could also have ILS with common inheritance that came up in Cecile's talk. Those are the two extremes of her correlated model, complete correlation and no correlation. Or we might just be willing to dismiss with ILS as a possibility and assume all our gene trees actually are displayed on the network. And then the final thing this question depends on is what data type we're going to deal with. And though I'm not going to be talking about genuine data at all in this talk, I'm going to be starting with gene trees typically. They might be metric or topological, and as the last talk made really clear, often one can do better with the topological information alone, that the metric information may be misleading due to various factors, including rate variation. The gene trees may be rooted or unrooted, or I may want to deal with some other summaries of the gene trees. And so while it may look like by summarizing I'm losing information, it may be desirable to get rid of that information, such as the metric information. This broad question of identifiability from biological data, the things that we can actually get out of sequences in some way, is largely understood only for level 1 networks. So just to remind you, level 1 means we have may have multiple cycles in the network, but they don't share any edges. Okay, so there's a level 1 network with 4 different cycles. Anything beyond that, very little is known. Okay, so the results that I'm going to outline, we consider the class of outer labeled planar networks. And while you may feel this isn't a very biologically defined network class, I should say no network class is very biologically defined. When we say level 1, it's just convenience because we have mathematical tools for understanding that. So this means we can have rather complicated blobs, lots of articulations, but we have to be able to draw the network in the plane so that no edges cross, and so that the taxa are on the outside. So to give you an idea of what the restriction is, whoops, that was not what I wanted to do. Somebody take out the pointer. Here we go. Let me go back. If, say, off of one of these edges in the middle here, I had, okay, we'll just go. If off of this edge I had a taxa coming down, then I would have to put in an edge crossing. So I can't have that situation. The taxa have to be on the outside when we draw it without any crossings. Okay. Okay, so I'm going to lay out 2 theorems. The first one is just a math one. The second one has to do with how you might do inference. So first of all, if I put in this condition that my networks have this outer labeled property, then around any of the blobs, there's a unique circular order. So for instance, you focus on this one, the most interesting one here. I have B and C coming off in one location. I move around to D, to E, to where F and G come off, and then up at the top, A comes off, and then I'm back around. And that, with any other drawing of that network, it may seem sort of obvious that we would have, again, that same circular ordering, but there are examples where it's not so clear. So, but there is a unique circular order. And the second fact is that if I can find some way of getting this circular order information from 4 taxon subnetworks, then I can find it for the large network. Okay, so what would be the implications of this in terms of inference? It's not saying I can determine which group is hybrid. Here E is the only hybrid, but there could be multiple hybrids coming off of this, and it's not telling me anything about how these smaller groups are separate later on. But here's the theorem, and this is going to be followed by an example 2 that perhaps illustrates that we cannot necessarily do that better. Okay, so this is actually 9 theorems. Because it depends on the tree model and the data type, and I've got 3 of each, so you can mix and match however you like. So this quartet information on circular order, and thus that for the full network blob, is identifiable for any of the 3 gene tree models I mentioned, so ILS with or without common inheritance. For the ILS alone, I have an extra technical condition I'm not going to go into, but ties into Hector's talk, or if the gene trees have to match those displayed in the network. And the 3 data types are average intertaxon distance from gene sequences. So what that means is I look at each gene, I see how far apart for each pair of taxa, the distance for that gene, and then after I've done that for every gene, I average. Okay. The second one is quartet concordance factors that have come up in a very large number of talks. So that is the quartet topologies of gene trees, the counts of them. And the third one is if I take all the genes and concatenate them, and I realize this seems strange because if you believe in the multi-species coalescence, you don't like to concatenate your genes. But if you concatenate them and compute the log dead distance, you can actually get the same quartet information from that. Okay, so this is, as far as we know, the first identifiability result for any kind of really complicated blob. Okay, and we've got lots of level 1 results, but beyond that, we don't really have much. But here's the counterexample that I think is also extremely important to think about. And in our paper, we have a number of these for the 9 different choices you can make and how you want to apply the theorem. So once we understand that, we can construct examples where we will not be able to tell networks apart based on a particular data type if it came from the model. So under the standard model of ILS, the network multi-species coalescent, if I have these two networks and I'm using quartet concordance factors, okay, so what goes into SNAP or Nanook for inference, these two networks can produce exactly the same concordance factors. Okay? So this is nice in level 1, but that means that if your data actually came from that network, you may think you have a level 1 network because it will exactly match what's going on. And obviously, I mean, we made that complicated, more complicated than you might want to think about for precisely the point of driving in. There may be some things you cannot find. Okay? And that green edge in particular, if we put it in, then that's not even planar anymore. Okay? So that means that we also will not be able to tell which networks are outer level planar. Okay? They can exactly mimic one that is. Where is that? Where are we with this? So at least with certain data types, there's going to be a limit to what we can infer. We don't actually know where that limit is now. We may have to settle for things like finding the order of things around the blob. Okay? You know, if I went in here and put another hybrid edge there, I could not check this, but that may well be indistinguishable from this one, from this data type, but then there would be 2 hybrids, and so I may not be able to tell how many hybrids I have. Okay? Even if you say there's 2 hybrids, well, I wouldn't actually be able to tell this was hybrid, because if I took that edge away, it's not. So, you know, we will simply have to get used to that. Now, it may be that if we go to more complicated data types, we can infer more, but there's logistical problems with that, computational problems. The computational time and difficulty in the algorithms is sort of preventing us from going further. Okay, so wrong way. That's the reference on the math archive right now, and I'll just stop there.
Hello, everyone. My name is Abigail Sequeira from the Huber Lab at Penn State, and today I will be discussing the development of a haplotype-based method to detect selective sweeps in ancient human genomic data. So to start out, let's first define what a selective sweep is. So sweeps describe the pattern in which a beneficial mutation rises to high frequency in a population. Typically, we have 2 types of sweeps, hard and soft. And hard sweeps result with a single haplotype rising to fixation or near fixation, whereas a soft sweep stems from standing variation or recurrent mutation and results in a few haplotypes rising to intermediate frequencies. There are many factors that can impact sweep behavior, such as recombination rates or selection strength. However, today we're going to be focusing on demography. So knowing the demography of your study system is really important because demographic changes are known to impact sweep detection. Bottlenecks increase the chance of inferring false sweeps. Admixture dilutes the sweep signal because it can cause a decrease in your sweep frequency in the admixed population. And this is especially important in humans because previous studies have reported that decrease in differentiation among populations over the past 10,000 years due to high levels of migration and admixture. However, ancient data allows us to look at sweeps before and after these demographic events and ameliorating a lot of these issues. So the majority of ancient data is pseudohaplotized, meaning that for every position of observed reads, we randomly select one of those reads, and that determines the genotype at that position. This technique helps to mitigate issues of low coverage and high missiness that is very typical in ancient data. Another method for dealing with these issues is imputation. However, imputation accuracy decreases with lower and lower coverage and as your samples become more diverged from your reference panel. So we are interested in using or developing a method that can fully utilize pseudohaploid data. In general, it's common practice to determine ancient sweeps based on the allele frequency distribution, but these methods can't accurately detect soft or partial sweeps. However, haplotype-based methods can. Haplotype-based methods have been rarely used on ancient data. To our knowledge, only 2 studies have used them. So in 2017, Martignano used the extended haplotype homozygosity statistic on imputed ancient data. And they used this to look at commonly known strong hard sweeps. However, they didn't do a genome-wide sweep analysis. More recently, Pandy et al. Modified the G12 statistic to work on simulated pseudohaplotized ancient human data, but they did not incorporate a spatial genomic component to their method, and previous studies have shown that including the spatial genomic signatures actually improves sweep detection. And so this is where we come in. So we are looking at this software called LASIP, which is a haplotype-based method that uses a likelihood model to determine the distortion in a haplotype frequency spectrum, or HFS. And then the Salty-Lassie method is specifically adds in this spatial genomic component. And this also was shown in this paper that it was originally published in to improve upon LASIP's sweep detection ability. So here, this figure here is showing the basic method of how Salty-Lassie works. So on the left, you have examples of what a distorted HFS would look like. So on top, you have a hard sweep where you would have a dominant haplotype, whereas with soft sweeps, you could have multiple dominant haplotypes. And so as the window moves further away from the sweep site in the genome, the HFS will slowly become more similar to that of the genome-wide frequency spectrum till it eventually just looks just like the genome-wide frequency spectrum. However, this method was only used on high coverage data and also data that had little to no missiness. And so we wanted to see how much we can really push this method using data that has low coverage and high missiness. And so this leads us to our main questions. So first, what are these ideal parameters for sweep detection using Salty-Lassie on ancient data? And then finally, wrapping back around to the demographic issue that I mentioned earlier, can we detect sweeps diluted by admixture? So for our methods, let's first break down the demography that we're using. So for this study, we simulate a realistic European demographic model, including population splits and admixture events over time. So our sweep starts, which are shown here in the center of the figure in red, introduce a mutation before and after these important population splits to simulate a mutation occurring in all 3 European ancestral populations, Anatolia, Western hunter-gatherer, and steppe, or just in Anatolia and Western hunter-gatherer, or just in Anatolia. All of these different times leads to different sweep dilutions in our admixed populations, Central Europe, Bronze Age, and our modern Europeans. The orange boxes represent the number of individuals that we sampled from these simulated populations and the time at which we sampled them. And then our sample numbers came from individuals that we had identified from the Allen Ancient Data Resource and how we grouped them together. Our basic protocol was to use an agent-based model program, SLIM, to conduct the simulations. We conducted neutral simulations and selection simulations using 3 selection coefficients of 0.01, 0.02, and 0.1, and had continuous and non-continuous selection on a beneficial mutation that's occurring in the center of a 5 megabase genomic region. We had 200 replicates for each population, for each sweep condition, and for our neutral simulations. After the simulations, we pseudo-haplotized only our ancient populations, and because ancient data is typically collected using a capture array, we simulated a standard ascertainment bias protocol on our populations. And then we added in missing data to the ancient populations that was based on our empirical missingness distribution. And then finally, we used Salty-Lassie on the simulated data to infer our sweeps. So Salty-Lassie allows for various user-defined parameters. We're interested in determining the ideal window size and measured in SNPs, and how many haplotypes or loci that we would need to filter out based on different missingness proportions. So we performed a window size comparison at 7, 51, and 101 SNPs, and we also compared different missingness filter parameters. It's important to note that these missingness filters are based on the proportion of missing data. So a missing filter of 0 means that if there is any missing data at that specific locus or within that haplotype, those are filtered out. Similarly, 0.1 means that if a locus or haplotype has over 10% missingness, those are filtered out. And the same can be applied for 0.2 and 0.3. So on to our results. So here, all the results that I'm showing are rock curves, and they have similar structure. But here I'm showing the strongest rock curves for the strongest selection coefficient of 0.1 and under non-continuous selection. So this means that selection stops after the admixture event. So along the x-axis, we have our false positive rate, or FPR, and the y-axis is the true positive rate, or TPR. And our simulations that have the most power, or can most accurately infer selection, will have a curve that has a low FPR but high TPR. So in this bottom center figure, there's this blue curve. So that shows it's high power because it's hugging along the y-axis and goes along the top of the x-axis. Going from left to right, our panels are showing our different window sizes, increasing from 7 to 101. And then going from top to bottom, we have our different sweep start times going further back in time as we go towards the bottom. So as we can see, power increases as we go further back in time, so that means older sweeps. And then we also increase power for the majority of our populations as we get smaller window sizes. In the case of continuous selection, meaning that selection is still occurring after our admixture event, we don't see much change in sweep detection power for our ancient populations. However, we do see an increase in the power to detect sweeps in our modern population. And also, just as a note, for our window size comparison, our missingness filters were the same of 0.3 for both haplotype and loci. Now moving on to how our missingness filters interact with the power to detect sweeps. And for these tests, we use a window size of 7, so because that smallest window size works best for the majority of our populations. And so again, here are our rock curves. And going from left to right, we have our different missingness filters and for all of the different tests that we ran. And as a reminder, these filter values are proportions. So if a haplotype or locus has above this missing filter, they're filtered out. And so as we can see here, different filter parameters are going to be working better or worse for different populations. And so what we are seeing here is that there's not one just standard set of missingness filters, but we still see an increase in power for the older sweeps. Now, in the case of continuous selection, we recover power in almost all cases. And for both continuous and non-continuous selection conditions, we find that the haplotype missingness filter seems to have the most impact on whether or not we actually are seeing power, especially in the case of continuous selection. And also possibly our most exciting result, and this is bringing back the demographic aspects and how admixture can dilute our detection signals. If you look, for example, at the top left corner, so we have two strong curves for our two admixed populations of modern Europeans and Central European Bronze Age, and we see that we retain very strong power for even the more recent sweep. And that, as a reminder, that sweep is only occurring in one of the three ancestral populations. And so in conclusion, as expected, continuous and stronger selection will improve the power to detect sweeps. And as I had just mentioned, the haplotype missing filter has the stronger effect on power, and this is most likely due to its direct impact that it has on estimating the sweep, more so than just filtering out a bunch of loci. And then finally, and and what I think is most important, if selection is continuous and strong enough, salty lassi can identify sweeps even in admixed populations and for more recent sweeps. So for future directions, we want to finalize this ideal set of salty lassi parameters for detection of hard sweeps. And then we also want to add in soft sweep simulations using this same realistic demography and use salty lassi to determine these soft sweep signatures. And then finally, we want to apply this method to empirical data to detect new signatures of sweeps previously obscured by admixture. And finally, I'd like to acknowledge everyone in the Huber Lab for their guidance and support, and our main contributor, Dr. Zachary Speech, in helping us develop this method, and then our funders of NIH and Penn State, and then finally, Evolution and all of you lovely folks here listening to my talk. Thank you. Right, we haven't looked at that yet, but based off of the empirical data, it's ranging. When I was looking at it, I think, if I remember correctly, some of the lowest coverage that we had was, I want to say, like 0.1, 0.5, so like below 1x. So we're talking like real low coverage. Yeah, in ancient data, how I see it, you're kind of lucky if you can get 1x for ancient data.
This is good, that's the range of my data.
Oh, that's great, yeah.
Yeah, thank you very much. Thank you, everybody, for staying here for the last talk of this session. So I'm not going to talk much about all the different features in SpitzTree, because that kind of makes for quite a boring talk. But there is a new version that's going to appear in press soon. And we've been working on this since the 90s, so there's about 180 algorithms in this software. And we're definitely not going to go through all of those. So I'm trying to do something different in this talk, so I asked myself, I spend all my time programming, I asked myself, what do you actually do with this program? What's it for, even? And so it's designed as an exploration tool. The idea is that you can go in and look at your data and look at it in many different ways and try and figure out what's going on in my data. And there's a particular focus in this software on phylogenetic networks, always has been from the get-go. And yeah, so can phylogenetic networks help me to understand my data? And so I actually asked ChatGPT, what do you think SpitzTree is for? And it said exactly the same. So this is new for me. I'm going to analyze some data. So I always heard about influenza, the influenza virus, that it's kind of cool because it's the genome's in 8 segments. And so here are the 8 segments. There's 1 gene per segment coding 1 or 2 proteins. And what's interesting about this in terms of evolution is there's something called reassortment. Different viruses infect the same cell and then the cell produces new viruses, but then it can mix and match different segments. So coming in, you've got uniform. Coming out, you've got a mixture. Okay, so one of the things you might want to use networks for is to try and explore this idea of reassortment. In particular, is there co-segregation? Are there segments that prefer to stay together, don't want to be reassorted, and other ones that don't mind if they get thrown in with some other segments? And can we use phylogenetic networks to help us to see this? Okay, so I've spent lots of hours of my life trying to find data sets to do stuff with. And this was actually, so it took me a while, but I found a really nice paper on the influenza virus. It's got data from the year 2005 to 2014, 14,000 strains, and it's published in eLife. And eLife is one of those journals that force the authors to make the data available. Often it's just, here are some accessions. I'm not going to give you anything that I've computed. Just you've got to go and do this all yourself. And so this is one of those papers where you can actually go in and say, okay, let's see the data that you've used. Makes your life easier as a methods developer. And so one of the things they looked at is, do segments tend to travel together through reassortment? And to do this, they built trees on the different segments, and then they computed the Robinson-Fultz distance. And so, yeah, that's a distance. The smaller the distance is, the more similar the topologies are. Okay, and so for this talk, I didn't use all 14,000 samples. I'm using 260 samples. So I looked for a year where there's not that many samples. So 2007, there's only 260 samples. And then for what I want to do, this is going to be about rooted networks. I want to be able to root stuff. So I selected an outgroup in a naive way. I just went for a different year. So these are all H3N2 subsamples of human-hosted viruses. There's all kinds of stuff in that data, but I stuck to human. And then I did, what you naively do. You align the sequences using MAFT, for example. Then I ran IQ-tree and let it do all its magic. I didn't select anything. And so I ended up with 8 trees, 8 gene segment trees, 261 taxa each. Okay, and so now we're going to see what can you do with, where does SplitStree come in? Well, you can load those trees into SplitStree. So the new version is not only for split networks like the old version, it's also there to let you work nicely with trees. So here are those 8 trees. If you can squint, or if you're sitting at the front, you can see that there's numbers on those edges. Those are bootstrap values. There's lots of edges in there with very low bootstrap values. I want to build networks on these trees, so that what I want to do is get rid of those edges. But before I do that, I just want to root. So in SplitStree, you can just say, you can select one or more taxa and say, this is the outgroup, and then reroute all the trees in that way. So you can root the trees by outgroup, and then you can say, okay, now collapse all the edges that have less than 80% bootstrap value. And now the fun begins. I go to that paper and say, okay, in that table where they compared the different genome segments, these two came out, you can't read that, I don't think, but these two came out the most similar. And the SplitStree, you can go into your list of trees and say, okay, these are the two I'm interested in, select those. And then what we want to do now is compute a rooted network. Now, today we heard computations on 20 taxa that take 2 days and so on. So that's not what you want if you want to interactively explore stuff. So here's a live demo of taking those 2 trees and finding a rooted network that contains them both with a minimum number of hybridization nodes. Press the button. Wait a minute. Press the button. Clock appears. And we're less than 5 seconds. And there should be a network there. Yeah, a network with hybridization number 9. So this network contains both those trees. And actually, the true minimal network here has hybridization number 8. So it's not perfect. But if you're a biologist who wants to explore the idea of using networks on your trees, it's a good start. And then you can go in there and say, let's have a look at stuff in more detail. You can go in there and try and figure out whether this group of strains here, whether you think reassortment is the reason why they are hanging off a hybridization node, or whether you think something else is going on. Yeah. So, yeah, let's look at 2 other ones. If I go back to that matrix, these 2, NP and NA, they're the second most similar trees. And if I run Phylofusion, I get a network containing both with 11, hybridization number 11. And now let's take the 2 that the least similar, the matrix segment and the non-structural segment. These seem to not care whether they reassort together or not. And now this algorithm, if I start the clock, run the algorithm, this is going to take longer. So the algorithm takes longer the more, the bigger the hybridization number is. So we have to wait. We have to wait 13 seconds. Okay. After 13 seconds, SplitStree figures out that for these 2 gene trees, you need 34 hybridization. The hybridization number is 34 to embed them together into a network. And so, as a biologist, you're probably not going to spend too much time looking at this particular network because it's quite complicated. And if I take like all the polymerase, the 4 polymerase segments, run the algorithm, then you get 66 hybridization. The hybridization number 66, so it becomes less interesting. But I guess that's what we all expected, how that only in special cases where you assume that gene trees are very similar, but there's some difference, only then you're going to be interested in looking at them in detail. But with this program, you can look at them, even stuff that is not worth looking at, but you can look at together. Okay, so we got this new method called phylofusion. It takes rooted trees as input, and now one of the nice features, it's got two really nice features. One is modifications are okay, so you can get rid of anything that you're unsure about ahead of time. You don't have to give it resolved trees, and also missing taxa are okay. You don't always have all taxa for all genes, so this algorithm can deal with that. It produces a rooted phylogenetic network, and it heuristically tries to minimize hybridization number. And the other nice thing about it is such a simple algorithm. So I'm going to show you for two trees. I think, so this is due to Lucien Zhang. He's explained it a number of times. I can never understand what it is, but it's actually so simple. You take two trees, and then you have to sort your taxa. So you order your taxa, and then you do a labeling of the internal nodes. And each taxon appears twice, once as a leaf and once as an internal node. And basically, to understand what those labels mean, if I go from you look at the right-hand side tree at the top, if I go from A to A, I pass two labels, E and B. These are the jumping off points where if I want to get to leaf E, I have to leave the path to A here, go down to E. And then if I want to get to B, I jump off at that position where there's a B. So it has a simple interpretation. I can't spend too much time talking about it. It's very simple, very easy to compute. And so for each sequence, for each taxon, you get a sequence. Okay? And so you get 5 sequences, because there's 5 taxa in both trees. And what do we do with sequences? We align them. So you just align for each taxon, you align the sequences. And so the set of sequences for a tree is an encoding of the tree. You can go from a tree to sequences, and from sequences to tree. And if you align those sequences, you get the encoding of a network. So you can just draw the network. Just by looking at the encoding before I even draw the network, I can already figure out the hybridization numbers too. It's an encoding, so it contains everything you want to know about it. Yeah. And so the computation is basically sequence alignment. For two sequences, if you've got two trees, each taxon, there's 2 sequences to align. That's just dynamic programming. If you've got more than 2, then we use the usual heuristic for multiple sequence alignment. And the other thing that I should mention is you have to look at all possible numberings of the taxa. So you've got a factor of n faculty there in theory. Yeah. And so we compared it against the only other algorithm that we know of that takes data with modifications and missing taxa, the autumn algorithm that Simone Lintz and I published a number of years ago. It's an exact algorithm. So as you see that, the dotted line is the number of hybridizations in the pair of trees that we're looking at. Autumn algorithm can only work on pairs of trees. It's an exact algorithm, so you can see those yellow dots on the dotted line where it gets exactly the right number of hybridization number equals the number of rooted SPRs that we put into the 2 input trees. But then at 60, you can only get up to 60 taxa, then the autumn algorithm just takes too long. But this algorithm keeps going. Even for 300 taxa, I think it took 100 seconds to run on 300 taxa with 30 rooted SPRs in the trees. Yeah, so that was something different. I'll try to show you how you could use a splits tree rather than going through all the features. And you can see that we're focusing on providing methods that we hope biologists can use, biologists can use to explore the idea of rooted phylogenetic networks on their gene tree data. Yeah, thank you, and thank my colleagues mentioned here.